Hello everyone and welcome back to the Clinical Cousins YouTube channel. Today we are going to go over excitation-contraction coupling in our skeletal muscle. Our muscle cells are invaginated with T-tubules. These are deep ravines that penetrate our muscle cells to make sure every cell has access to the proper electrolytes. Now we will learn how the muscle contracts. As we know, the acetylcholine has been released and is now bound to our nicotinic receptor on our muscle cell membrane. This causes sodium and potassium channels to open, which bring our voltage-gated sodium channels to threshold potential. This action potential is propagated down the sarcolemma, or muscle cell membrane, until it reaches what is called a dihydropyridine receptor, or DHP receptor. This DHP receptor is what is known as a voltage-gated L-type calcium channel. When our DHP receptor is brought up to threshold, it will allow a small amount of calcium inside of our muscle cell that we call trigger calcium. It is called trigger calcium because this seemingly tiny amount of calcium causes our ryanodine receptor, which is located on our calcium-stuffed SR, or sarcoplasmic reticulum, to open. Because the ryanodine receptor has opened the SR, the inside of our muscle cell receives a calcium shower. Because there is plenty of calcium inside of our cell, some of this calcium is showered on our troponin, specifically our troponin C. As soon as this calcium binds to troponin C, the troponin complex undergoes a physical change and removes the tropomyosin cover off of our globular actin. This allows the thick myosin heads to finally bind with the thin globular actin. The second that our myosin and actin bind with each other, we say that a cross bridge has formed. It is important to note that myosin will use ATP energy to go from actin to actin to actin until there are no more thin globular actins left to bind with. Also, this process will cease when there is a low amount of calcium inside of our cell. This occurs because once our motor neuron stops firing, this entire process will be reversed. There will be no action potential, trigger calcium, calcium shower, or calcium binding to troponin C. At this point, troponin and tropomyosin will move back to cover the thin globular actin, and the cross-bridge cycling will cease. Now, what has just happened? Our action potential in our motor neuron has caused an action potential in our skeletal muscle, which has stimulated our myosin to walk along the globular actin, which shortens our muscle and generates force. At the end of our nice play, we must remember to thank the ones who clean up. Our calcium shower is constantly being cleaned up by our nice calcium ATPase on our SR. This calcium ATPase is constantly shoving the calcium back inside of our SR so that we are ready to contract our muscle again. As always, thank you for taking the time to learn with us today, and remember to like and subscribe for more content.